Hi, I'm Aurel Enriquez, and this video contains our summarized review discussion in Microbiology 2, Mycology, and Virology. Although, in comparison to our previous summarized review discussions, where we simply speed run through all of the topics that we've studied in one semester, dito po magkakaroon tayo ng practice questions. So, I'll be showing you an easy practice question with multiple choices, and you can pause the video to try and answer, tapos i-play nyo na lang po ulit para malaman yung tamang sagot and to watch the rationale or explanation after each questions, okay? This discussion will be divided into two parts. So for this video, which is the first part, we'll be having a review in mycology. Let's begin. First question, mucorellus members, referred to as mucormycetes, typically show which of the following hyphae morphology? The correct answer is rhizoids. Rhizoids are root-looking structures that are seen in some mucorellus members. So as we can see right here in this illustration of the Rhizopus stolonifer species, ito po yung mga rhizoids, and they are connected together by a running hyphae referred to as a stolon. So this photo right here is an actual sample of the Rhizopus stolonifer. So kung saan po makikita natin nandito yung sporangiophores nila, no, they're growing upwards. And then on top of that, makikita po natin na nandito yung sporangiospores. Okay, nandito po sa taas yung sporangiospores nila. And then we can't see all of them dahil nakabaon sila, but right here, no, nandito po, nakikita natin na, ayun nga, they do have these kind of like root-shaped or root-looking structures, and those are the rhizoids. Okay, so they kind of look like yung mga maliliit na puno, diba, na may ugat sa ilalim. Okay, so again, those are the rhizoids of mucorellus members. Next question, which of the following photos show an example of a favic chandelier? The correct answer is letter C. The favic chandelier is also known as the antler hyphae dahil kahawig daw po nila yung mga sungay or antlers of moose, as we can see right here in this photo. Okay, so let's just quickly talk about all the other structures mentioned here. Okay, so next is the racket hyphae, where they have enlarged or club-shaped areas. Okay, so tulad ng isang badminton racket or tennis racket, diba? May mga areas po na diretso lang, tas pahaba sila, and then randomly at some point of the hyphae, there are areas that seem enlarged or kind of like balloon-like in appearance. So ayun nga po, no, that's the racket hyphae. Next, we have the spiral hyphae, where we could easily observe that there are tight coils throughout the structure. Next are pectinate bodies, which have been described as comb-like structures. So nakikita po natin, no, across the hyphae, may mga extensions din po siya, giving it its comb-like appearance. Next are rhizoids or the root-like structures. Ito po napag-usapan na natin kanina. We usually see this among mucorellus members. And ayun nga po, they do look like little roots. Okay, so ayun po. People might be asking, or some people might be confused, no? How do we differentiate the favic chandelier or the antler hyphae in comparison to pectinate bodies? Because for some people, this might look a bit confusing because they both have branches. Now, again, looking at the definition for the antler hyphae, they only have branching at the tips. Okay, so kung titignan natin tong picture na to, diba? Kung ito yung kumbaga parang length ng hyphae, nandito lang sa pinakadulo yung branches or extensions, diba? Let's compare that to the pectinate body, which is right here. Hindi na confined sa dulo lang ng hyphae yung branches or extensions, diba? As we can see, at the other areas of the hyphae, may mga parang branching or extensions din siya. Hindi lang dun sa pinakadulo. Okay? So when you're looking at the antler hyphae, the branching are only at the tips. But if you're looking at a pectinate body, there may be extensions or branches and everywhere else. No? Everywhere else on the hyphae. So, based on the previous explanation, let's now answer this next question. Which of the following photos show an example of pectinate bodies? The correct answer is letter B. Okay, so ayan po, no, i-compare natin siya dito sa mga photos or examples na katabi niya. So example A and example C are both photos of the antler hyphae or fabric chandelier, diba? Tignan po natin yung letter A, diba? So in this photo, kung ito yung hyphae, I hope you're seeing my pointer. Kung ito po yung hyphae, diba? Yung branches niya nandito lang sa pinakadulo, 'di ba? Wala kang nakikitang branches anywhere else, all right? An example is this one right here. Also for letter C, 'di ba? As we can see here in this example, if this is the hyphae, no, most of the branches, no, are right here at the tips, 'di ba? There's a concentration of branches or extensions dito lang sa dulo ng hyphae. But if we look right here in the photo in the middle or yung option B, makikita nyo po na kung ito yung hyphae, no, this one right here, we're seeing that there are extensions, no, throughout the hyphae. Hindi siya nakakoncentrate lang dun sa pinakadulo. Okay? So again, which of these following photos would show an example of a pectinate body? The correct answer here is letter B. Next question, if a fungal species could only reproduce by forming ascospores, this would be classified as which of the following? The correct answer is teleomorph. So let's first answer the next two questions before we proceed to discuss the answers. Next question, if a fungal species could reproduce by forming a macroconidia, microconidia, and basidiospore, this would be classified as which of the following? The correct answer is cynanomorph. Next question. If a fungal species could reproduce by forming an arthroconidia and a zygospore, this would be classified as which of the following? The answer is an anamorph. So let's now discuss the answers. Let's now discuss the terminologies that are used in describing fungi that could undergo sexual reproduction. So if you would remember from past discussions, the structures that are associated with the sexual reproduction of fungi would include yung ABZ, diba? Ascospores, basidiospores, and zygospores, right? Next, let's also make a list of the structures that are associated with the asexual reproduction of fungi. So it would be the annelids, phyalides, arthroconidia, and yung macro and microconidia. Diba po? Okay, so ayan po. So let's talk about these terminologies that we have right here. First up is a teleomorph. So a teleomorph is a fungi that reproduces sexually. So kapag sinabi nating teleomorph ang isang fungal organism, ano yung mga structures na kaya niyang i-produce? Yung ABZ, diba? Ascospores, basidiospores, and zygospores. Next is an anamorph. So when we say anamorph, it's a teleomorph, so it's a sexually reproducing fungi that can also reproduce asexually. So let's say, for example, this fungi produces an ascospore. All right, this fungi produces an ascospore, but at the same time, it could also produce arthroconidia. All right, so yan po, that would be classified as an anamorph. Okay, next is a cynanomorph. So a cynanomorph is a fungi that has more than one anamorph. Okay, more than one daw po yung asexually reproducing structure niya, and that is present for the same teleomorph. Okay, so mayroon daw po siyang isang sexually reproducing structure at dalawa or more than one na asexually reproducing structure. So let's say, for example, this organism produces a basidiospore. So yun yung teleomorph niya. But it's also capable of producing a macroconidia and a microconidia. So yun yung dalawang asexually reproducing structure niya. So now that organism would be classified as a cynanomorph. Okay, so I hope you understood that. Next question. Which form of sporotrichosis produces nodular ulcerative lesions along the patient's lymph channels? Your choices are lymphocutaneous, lymphatic, fixed cutaneous, subcutaneous, or lymphonodular. The correct answer is lymphocutaneous. Okay, so as you can see right here, listed here are the different clinical manifestations of sporotrichosis. We have lymphocutaneous, fixed cutaneous, mucocutaneous, and pulmonary sporotrichosis. So for this discussion, we'll focus on these two right here. So first up is lymphocutaneous sporotrichosis, wherein we would be able to observe nodular ulcerative lesions along the patient's lymph channels. So if we look at this set of photos right here, makikita po natin na kung example, ang point of entry ng organism is dun sa hands lang ng patient, ma-observe natin na the lesions start off at the hand, but they eventually move up towards yun nga kung nasan yung lymph channels ng pasyente. So it starts spreading throughout the patient's arm. Okay, but let's compare that to fixed cutaneous sporotrichosis, wherein the lesions are confined at the site of the inoculation. So if we look at these photos right here, we would be able to observe na ayun nga po na kung nasaan lang yung point of entry no organism, then doon lang din natin ma-observe yung clinical manifestations nito, or doon lang natin ma-observe yung lesions that were formed during the course of the infection. All right, because if you would look over here dito sa mode of transmission, diba? Mode of transmission would include occupational exposure, diba? Exposure to rose thorns or sphagnum moss. So if the fungal cells get inoculated onto the patient's skin or subcutaneous tissue, dun po magsisimula yung infection. Next question. Which of the following is the causative agent of South American blastomycosis? Our choices are Agelomyces dermatitis, Coccidioides immitis, Penicillium barnefei, Coccidioides possidacei, and Paracoccidioides brasiliensis. The correct answer is Paracoccidioides brasiliensis. Paracoccidioides brasiliensis is the causative agent of the actual term is paracoccidioidomycosis, but listed here are all its other names. So this could also be referred to as paracoccidioidal granuloma, the Lutz Splendor Almeida disease, the South American blastomycosis, or the Brazilian blastomycosis. So pinakakilala nga siya, no, as a causative agent of South American blastomycosis. So how do we recognize Paracoccidioides brasiliensis in the laboratory? As we can see right here in these photos, no, since Paracoccidioides brasiliensis is a dimorphic fungi, side note lang, lahat po ng causative agents ng systemic fungal infection, they are dimorphic. Okay? So, lahat ng causative agents ng systemic fungal infection ay mayroong mold form at mayroong yeast form. So, going back to Paracoccidioides brasiliensis, its mold form has been described to be they appear like one-cell chlamydoconidia, and they look very similar, indistinguishable with blastomyces dermatitis, as we can see right here in this photo. So we recognize them by the appearance of their yeast form. Okay, so their yeast form are seen as multipolar budding yeast cells with a narrow-based attachment. So pag sinabi nating multipolar, the mother cell is capable of producing daughter cells at multiple different directions. Pag sinabi naman nating narrow-based attachment, it means that sobrang nipis, no? Manipis lang yung point of attachment between the mother cell and the daughter cell. So in some references, this has been given the description of a Mickey Mouse cap or mariner's wheel, as we can see right here in these examples. Next question. Which of the following is the causative agent of North American blastomycosis? Our choices are Paracoccidioides brasiliensis, Blastomyces dermatitis, Penicillium barnefei, Coccidioides immitis, or Geotrichum candidum. The correct answer is blastomyces dermatitis. Okay, so here are the other names for the disease. It's the Gilchrist disease, the Chicago disease, and ayun nga po, we know it as the North American blastomycosis. So ayun po no, I hope na eliminate nyo na yung isa sa mga choices kasi na-discuss na natin that Paracoccidioides brasiliensis is the causative agent of South American blastomycosis. So let's look at how we would recognize blastomyces dermatitis in the laboratory. As previously mentioned, lahat ng causative agents ng systemic fungal infections ay dimorphic, diba? So ang blastomyces dermatitis, it also has its mold form and its yeast form. So its mold form are seen as oval or globose conidia that grows out of very short conidiophores, as we can see right here in this photo. So tama nga naman yung description kanina, no? It's quite difficult to differentiate them in comparison to Paracoccidioides brasiliensis. So let's look at its yeast form. All right, so its yeast form are large and they have broad-based budding. Okay, they have large yeast cells that are unipolar. No, they are unipolar and they have a broad-based budding. So ano po ba ang meaning nun? Pag sinabi natin unipolar, if we look at this photo right here, they're only capable of producing one daughter cell in one specific direction. Hindi katulad ng Paracoccidioides na maraming daughter cells yung kaya niyang i-produce at multiple different directions, diba? So that's what we mean when we say unipolar. Next, when we say broad-based budding, malapad daw po, no? It has a broad point of attachment between the mother cell and the daughter cell, as we can see right here. So that's for blastomyces dermatitis. Next question. Which fungal pathogen can produce large, round, echinolate macroconidia, as we can see right here in this photo? Our choices are Histoplasma capsulatum, Paracoccidioides brasiliensis, Coccidioides immitis, Blastomyces dermatitis, or none of these are correct. The answer is Histoplasma capsulatum. So, I hope you were able to eliminate the other choices na po, no? Kasi na-discuss na natin yung Blastomyces dermatitis and yung Paracoccidioides brasiliensis. So, let's look into the mold and yeast form of Histoplasma capsulatum. Uulitin ko lang po, no, just to establish that fact, no, that the causative agents of systemic fungal infections are dimorphic. Kaya mayroon siyang dalawang forms. So the mold form of Histoplasma capsulatum is seen as round, large, and they have an echinolate or tuberculate macroconidia. When we say echinolate or tuberculate, they have these rough or spiky projections on the outside or on the surface of the cell. So shown here are the different examples. So this photo right here and ito po, those are the mold forms of Histoplasma capsulatum. Let's compare that to its yeast form. Its yeast form has been described to be small, oval, and encapsulated yeast cells that are seen within monocytes or macrophages. Now, being encapsulated is actually a misnomer, okay? Ang pangalan niya ay Histoplasma capsulatum, but it doesn't actually have a capsule, all right, because the appearance of being encapsulated is only a staining artifact, all right. Their capsules are only artifactual because their yeast cells eventually shrink during the staining process. So when the yeast cells shrink, a space surrounding them is left behind. So on microscopic examination, they appear like they do have capsules when they actually don't. That's just an airspace that has been formed when the cells were shrinking. Okay, so that's the yeast form of Histoplasma capsulatum. But again, on the previous question, we were shown the mold form of Histoplasma. Next question, identify the instrument or alternative diagnostic method that was used in this photo. Our choices are Wood's lamp, fluorescence microscopy, immunofluorescence, or none of these. The correct answer is the Woods lamp. So the Woods lamp, this is an alternative method for the diagnosis of superficial and even cutaneous fungal infections. At kasama na nga po doon yung Tinea versicolor, which is caused by Malassezia fur. So as we can see right here, we could make use of the Woods lamp, which is a form of UV light. All right, the Woods lamp is a form of UV light. We could point that at the area where we suspect that the infection is currently happening, and we would be able to visualize these kind of like white or light blue fluorescent areas. So those are the areas of infection. So most likely po kung magko-collect ka ng sample or skin scrapings, yan po yung tamang lugar para mag-collect ng sample. Okay, so that's for the observation of Tinea versicolor using the Woods lamp. Next question. Based on the site of infection, identify the type of cutaneous mycosis shown here. Our choices are Tinea corporis, Tinea manum, Tinea imbricata, Tinea versicolor, or Tinea nigra. The correct answer is Tinea manum. So as we can see right here on this list, ang mga cutaneous fungal infections po kasi, they are named with the word Tinea first and then the affected body site. All right, so in this list, we can see the different forms of infection and their locations. So Tinea favosa doesn't actually follow this pattern. Tinea favosa is named after the microscopic appearance of its causative agent. All right, so Tinea favosa and Tinea capitis, they are both scalp infections. Tinea barbae, is an infection of the beard area. Next is Tinea manum, an infection on the hands. Tinea corporis is a fungal infection on the body. So sa torso, kasama na rin po yung arms and legs. Okay, on the arms and legs, that's also considered as Tinea corporis. Tinea cruris is an infection on the groin or inguinal area. In English is otherwise referred to as jock itch or hadhad in Filipino. Next is Tinea pedis, which would infect the feet. So in English, that's referred to as athlete's foot, and in Filipino, it's known as alipunga. Okay, next is Tinea unguum. This would infect the nail areas or our nails. Okay, so either on the hands or on the feet. Or kung nasaan ka pa man mayroong kuko. Next question. What is the specimen of choice for the diagnosis of Tinea versicolor? Our choices are skin biopsy, skin scrapings, hair clippings, or nail clippings. The answer is skin scrapings. So, as we've mentioned kanina po sa previous question, Tinea versicolor or pityriasis versicolor is caused by Malassezia fur. Now, the preferred specimen or specimen of choice for the diagnosis of this disease are skin scrapings. Okay, so either you perform microscopic examination or ayan nga po, the usage of the Woods lamp. Now, if you were to perform microscopic examination, you could use the LPCV or lactophenol cotton blue staining, and you should be able to observe abundant septate and branched hyphae plus clusters of budding yeasts. Okay, so ayun nga po, the combination of the budding yeast cells and the bunch of septate branched hyphae gives them what has been known as the appearance of the spaghetti and meatballs, as we can see right here in this photo. Okay, so ayan po. That's for Malassezia fur. Again, the specimen of choice for Tinea versicolor is skin scrapings. Next question, identify the causative agent of cutaneous mycosis shown here in this photo. Our choices are Trichophyton mentagrophytes, Trichophyton schornleinii, Microsporum arduinii, Nanitzia gypsea, or Hortea werneckii. The correct answer is it's Trichophyton schoenleinii. So on the previous photo, we were shown an example of the Favic chandelier or the moose antler hyphae, diba po? So we were looking at a fungal element kung saan po may branches dun sa tips ng hyphae. And again, that's an example of the Favic chandelier or the moose antler hyphae, which is specifically produced by Trichophyton schoenleinii and is the causative agent of Tinea favosa or Tinea favus. Kaya nga po, di ba, compared to the other forms of cutaneous fungal infections, hindi siya pinangalanan based on the infected site, diba? Ang Tinea favosa or Tinea favus po, it's named after the appearance, the microscopic morphology of the causative agent. Alright, so again, Trichophyton schoenleinii is the causative agent of Tinea favus, wherein microscopically we would be able to observe the moose antler hyphae. Next question, what is the causative agent of the cutaneous infection shown here? The correct answer is Trichophyton concentricum. Now, before we discuss, let's answer the next question. Next, based on the appearance of the lesions, identify the form of cutaneous mycosis shown here. The correct answer is Tinea imbricata. Okay, so emphasis on the appearance of the lesions. Okay, hindi po location ang tinitignan natin dito. Now, if the question is asking you based on the location or based on the site of the lesions, identify the form of cutaneous mycosis shown here, then ang dapat nating isagot ay Tinea corporis kasi 'di ba nasa arm ng pasyente. Kung site or location po yung tinitignan na description, okay? But in this case, yung appearance po ng lesions yung pinapa-observe sa atin. So again, the correct answer is Tinea imbricata. So as you can see right here, Tinea imbricata is classified as a cutaneous fungal infection, and this is caused by Trichophyton concentricum, which is quite easy to remember because they're capable of producing concentric rings of scaling tissue. Let's look at this photo right here. Parang may pattern na paulit-ulit na pinoproduce yung Trichophyton concentricum, di ba? Parang may sinusundan lang sila na magkakaduktong na pattern until, ayun nga po, ma-observe natin na may isang malaking lesion na dun sa body ng pasyente. So again, that is referred to as Tinea imbricata. So the disease is Tinea imbricata. The causative agent is Trichophyton concentricum. And the clinical manifestation is concentric scaling tissue. Next question, which form of Tinea capitis is characterized by an ectothrix hair involvement? Our choices are black dot ringworm, gray patch ringworm, black piedra, or white piedra. The correct answer is the gray patch ringworm. So let's look at this slide right here. When we say ectothrix hair involvement, the fungal growth is only observed on the outside of the hair strand or hair shaft, as we can see right here in this illustration. But when we say endothrix hair involvement, it means that the fungal organism is capable of growing on the inside of the hair shaft, okay? But in the previous question, alin nga daw po doon yung ectothrix lang yung infection na kayang idulot, and the correct answer to that is the gray patch ringworm. But gray patch ringworm and black dot ringworm are both classified as Tinea capitis, which is a form of fungal scalp infection. Also, wag din po kakalimutan yung mga causative agents nila. So, for the gray patch ringworm, this is caused by itong dalawang microsporum, diba? Microsporum AF, Microsporum AF, Microsporum audrinii, and ferruginea. Next, for the black dot ringworm, its causative agents are yung dalawang Trichophyton, diba? Trichophyton TV, right? Trichophyton tonsurans and violation. Next question. Identify the causative agent of cutaneous mycosis shown here. Epidermophyton flucosum, Trichophyton schoenleinii, Microsporum arduinii, Microsporum canis, or Microsporum gypsum. The correct answer is Microsporum canis. So if we were to observe the macroconidia of Microsporum canis, they have been described to be spindle-shaped with tapered ends. Medyo pahaba daw po sila, tapos matalim or patusok yung magkabilang dulo. That's what we mean when we say tapered ends, as we can see right here in these photos. So again, that is for Microsporum canis. Next question. Which form of cutaneous mycosis is characterized by the formation of crusty, cup-shaped flakes on the scalp, which may lead to hair loss and scar tissue formation? Our choices are Tinea capitis, Corporis favosa, or Cruris. The correct answer is Tinea favosa. So we've already talked about this in the previous question, di ba po? So as we can see right here, the clinical manifestations of Tinea favosa or Tinea favus would be, ayan po, there's an accumulation of fungal mycelia or excess fungal mycelia and the dead epithelial cells on the skin or scalp of the patient, leading to the formation of those crusty lesions. So those crusty or cup-shaped flakes or lesions would now be referred to as a scutula. Okay, ulitin po natin, no? Anong tawag dun sa mga crusty cup-shaped flakes? Sa case ng Tinea favus, it's scutula. Saan gawa yung scutula? Fungal mycelia and dead epithelial cells. So again, this is something that you would be able to observe if the patient has Tinea favosa or Tinea favus. And again, its causative agent is Trichophyton schoenleinii. Next question. Identify the causative agent of cutaneous mycosis shown here. Our choices are Microsporum canis, Nanatzia gypsea, Epidermophyton flucosum, Microsporum arduinii, or Trichophyton tonsurans. The correct answer is Nanatzia gypsea, which is formerly known as Microsporum gypsum. So if we were to observe the macroconidia of Nanatzia gypsea, we would be able to observe that they are much thicker, and they've been described to be cigar-shaped. When we refer to cigar-shaped, they kind of have like this very thick tobacco-like appearance, and they have rounded ends. So if we were to compare that with Microsporum canis, yung magkabilang dulo po ng Nanatzia gypsea, hindi sila ganun katalim. Hindi sila patusok, diba? They are much more blunt and rounded. Okay, so that's for the macroconidia of Nanatzia gypsea. Next question. Which type of subcutaneous mycosis would we be able to observe these structures? The answer is chromoblastomycosis. Sagutin po muna natin yung susunod na tanong before we discuss. Next question. What do we call these structures? The correct answer is medlar bodies. So let's now talk about chromoblastomycosis. Chromoblastomycosis is otherwise referred to as verrucous dermatitis or chromomycosis. Okay, so as we can see right here, this is a chronic infection of the skin and subcutaneous tissues, which is why nakalista na po ang chromoblastomycosis as a form of subcutaneous fungal infection. So usually this is confined to the extremities like the hands or legs or feet as a result of traumatic puncture or accidental exposure or maybe occupational exposure. So we would be able to recognize a case of chromoblastomycosis by observing cauliflower-like lesions, as we can see right here on these photos. Ayan po, may cauliflower-like or cauliflower appearing lesions yung pasyente sa arms and hands nila. Okay, so just a side note, not just a reviewer. For the diagnosis of subcutaneous fungal infections, we usually prefer direct tissue examination. Diba? Ulitin po natin, the preferred method of diagnosis for subcutaneous fungal infections, which would include chromoblastomycosis, is direct tissue examination. So dito po, sa case na to, dito sa mga kung nasaan yung cauliflower lesions, that is where you should be collecting your sample. Once you collect your sample, you observe that microscopically, and for chromoblastomycosis, we should be able to observe the sclerotic bodies, as we can see right here. So sclerotic bodies are also otherwise referred to as medlar bodies or the copper penny bodies. All right, so diba, that's our triple C, diba, CCC, diba, chromoblastomycosis, producing cauliflower lesions with the copper penny bodies. Okay, so again, that's for the the copper penny bodies of chromoblastomycosis. Next question. Which form of subcutaneous mycosis is usually caused by darkly pigmented fungi? Our choices are actinomycotic mycetoma, chromoblastomycosis, phaeohyphomycosis, sporotrichosis, or eumycotic mycetoma. The correct answer is subcutaneous phaeohyphomycosis. Okay, so let's kind of like break down that question. No, which of the following diseases is caused by darkly pigmented fungi? Una sa lahat, ano ba ang tawag natin sa mga darkly pigmented fungi? We refer to them as demaciaceous or phaeoid fungi. Okay, so it would make sense that the disease associated is phaeohyphomycosis. Alright? So ayan po, no, they're caused by darkly pigmented fungi, and we would be able to observe yeast-like cells within tissue samples as shown right here. Okay? So again, the preferred method of diagnosis for subcutaneous fungal infections like these ones is direct tissue examination. So kung nasaan nyo po nakikita itong mga bukol-bukol sa ilalim ng balat ng pasyente, doon po tayo kukuha ng tissue sample. And of course, you already know this, the causative agents of phaeohyphomycosis is A, B, C, and E. Diba? Alternaria, bipolaris, curvularia, and exophiala species. Next question. What would you call the telomorph structure of Pseudoaliscaria boydii? Our choices are perithesia, ascogonia, antheridia, apothesia, or cleistothesia. The correct answer is cleistothesia. So as we can see right here, Pseudoaliscaria boydii is a causative agent of eumycotic mycetoma, and it's capable of producing two different forms. It can produce an anamorph and a teleomorph. Okay, so for this one, its anamorphic form is referred to as the Pseudosporium boydii, where we would be able to observe oval conidia at the tips of annelids. Okay, so let's look at this example right here. Yung mga pahaba na structure po na nakikita nating nag-extend from the hyphae, ito yung hyphae. Okay, itong mga structure na to, ito po yung mga annelids. Okay, and then those oval structures na nakikita nating nakadikit sa kanila, yun po yung conidia or yun yung annelids ng Pseudosporium boydii. But if we're looking at the teleomorph form of this same organism, then ang itatawag na natin sa kanya ay Pseudoaliscaria boydii, wherein it's capable of producing what is known as a cleistothesia. So a cleistothesia is a structure that contains spores on the inside. Now, if you would remember from the previous question, no, when we say an organism is a teleomorph, it's capable of reproducing sexually, diba? Ano nga ulit yung structures associated with sexual reproduction? ABZ, diba? Ascospores, basidiospores, and zygospores. So looking at the cleistothesia and understanding that it has a container on the outside and spores on the inside, alin siya doon sa ABC? It's an ascospore, diba? The Cleistothesia is just another form of an ascospore, or the Cleistothesia is an ascospore that is produced by the Pseudoaliscaria boydii. So again, ang general structure kasi ng mga ascospores ay container sa labas, tapos spores sa looban. Alright, so again, the Cleistothesia is just a form of ascospore. Next question, which type of subcutaneous mycosis would we be able to observe these structures? Our choices are chromoblastomycosis, sporotrichosis, blastomycosis, phaeohyphomycosis, or eumycotic mycetoma. The correct answer is sporotrichosis. Now, before we discuss, let's first answer the next few questions. Next, what do we call these structures? Our choices are asteroid bodies, sclerotic bodies, medullary bodies, or encapsulated yeast cells. The correct answer is asteroid bodies. Next question, on which cells would we be able to find these structures? Our choices are macrophages, lymphocytes, monocytes, or neutrophils. The correct answer is neutrophils. Let's discuss the answer. Now, the structure that was shown to us in the previous questions is the asteroid bodies, or otherwise referred to as the Splendor-Hopley phenomenon. So the asteroid bodies are usually found within polymorphonuclear cells, or we more commonly know them as neutrophils, di ba po? So dun sa mga neutrophils, meron silang radiating filamentous structures or filamentous extensions. Ano ba ang itsura nun? So let's say for example, ito yung neutrophil ng pasyente, tapos nasa gitna yung nucleus nila, di ba? Eventually, no, in cases of sporotrichosis, no, these filamentous structures would be formed. So may mga guhit-guhit po tayong makikita mula dun sa center ng neutrophil moving to the outside, or kind of like connected to the outside, as we can see right here. So mula dito, nag-extend to the outside yung filaments. Okay, so again, this one specimen right here, or this one structure right here, is referred to as asteroid bodies. In this photo, there are two of them. Okay, so those are the asteroid bodies that are observed in cases of sporotrichosis. Okay, keep in mind though that there may be other diseases that is associated with the formation of asteroid bodies. Okay, the formation of asteroid bodies, or the Splendor-Hopley phenomenon, is not exclusive to sporotrichosis. There may be other diseases out there kung saan makakapag-observe din tayo ng mga asteroid bodies. Okay? Next question. Which of the following is the correct microscopic appearance of the mold form of the Sporothrix species? The correct answer is letter A. So as you can see right here, ito po yung mold form ng Sporothrix Schenckii complex, wherein they're capable of producing conidia that is arranged in a rosette or flower-like formation. Okay? So again, ito po, no, rosette or flower-like formation, which is kind of like an interesting coincidence dahil ano pong disease associated sa Sporothrix Schenckii species? Sporotrichosis, diba? Which is otherwise referred to as the Rose Handler's disease, di ba po? Tapos on microscopic morphology or microscopic observation, they're also capable of producing conidia that is arranged in a rose or flower-like pattern, di ba? So again, the mold form, no? Yung mold form po ng Sporothrix species is, ayan po, conidia that are arranged in a rosette or flower-like formation. Yung picture po dito sa letter B that's the yeast form. Yeast form na po yan ng Sporothrix species. And yung letter C, nakita na po natin yan sa previous question. Those are the asteroid bodies. Next question. Which of the following is the correct microscopic appearance of the Bipolaris species? The correct answer is letter C. All right. So again, bipolaris species is one of the causative agents of phaeohyphomycosis. Ulitin natin, the causative agents of phaeohyphomycosis are A, B, C, and E. Diba? Alternaria, bipolaris, curvularia, and exophiala species. Now, in the previous question, yung option A po, that is curvularia. It has a crescent-shaped conidia. Yung letter B naman po dun sa previous choices natin, that's an example of alternaria where they are muriform and tapered. When we say muriform and tapered, yung isang dulo niya, matalim or patusok. That's when we say muriform, it has these cross walls or divisions that are parallel to each other, 'di ba? So parang may pa-diagonal, may pa-vertical na hati-hati dun sa looban ng cell. But as we can see right here, for the bipolaris species, they're capable of producing, ayan po, multi-celled or multicellular oblong or cylindrical conidia. So again, the correct answer to the previous question is letter C. Next question, which constitutive agent of chromoblastomycosis is described to have erect conidiophores and elliptical-shaped conidia only at the upper portion near the tip, as we can see here in this photo? Our choices are Fusarium falciforme, Rhinocladiella aquaspersa, Cladophyalophora carionii, Fonsicea petrosoi, and Phyalophora vericosa. The correct answer is Rhinocladiella aquaspersa. So let's look at these structures right here. So we recognize the presence of Rhinocladiella aquaspersa by the observation of erect conidiophores or straight up conidiophores, as we can see right here within the yellow marker. Next, they have conidia that is only concentrated at the upper portion near the tip of those conidiophores, as we can see right here inside of the orange marker. Next, with regards to the shape of their conidia, they've been described to be elliptic. Diba? Parang pahaba daw po yung itsura ng conidia ng Rhinocladiella aquaspersa, which to me, they kind of look like grains of rice. Diba? So pag ganun po yung itsura, we're looking at Rhinocladiella aquaspersa. Next question, which of the following is not one of the known agents of rhinocerebral mucormycosis? The correct answer is letter A, dahil ang letter A po is Aspergillus, specifically Aspergillus niger. So as we can see right here, nandito po yung iba't ibang appearance or macroscopic and microscopic morphology ng 4 Aspergillus species associated with human infections. Now, it's quite difficult to identify Aspergillus species based only on microscopic appearance, which is why we have to also look into their macroscopic appearance or colony morphology for us to be able to identify them or differentiate them. Aspergillus niger is the easiest one to recognize dahil kung anong itsura niya on microscopic appearance, yun din yung itsura niya on colony morphology, diba? It has a kind of like brown appearance. Let's compare that to the others. Aspergillus fumigatus has a dark green colony on the inside with a white outer edge. Next, for Aspergillus flavus, they've been known to produce bright yellow or yellow-green colonies. Next one po is Aspergillus terreus. Their colony morphology has been described to be kind of like dull pink or dusty pink in appearance. Okay, but microscopically, if you're new to the observation of Aspergillus species, this might be difficult to recognize. Next question, which of the following is known as the black bread mold? The correct answer is letter A. So yung letter A po, that's the Rhizopus species. Yung letter B po, that's an example of Oreobasidium. And yung letter C po is, again, the photo of Aspergillus niger. So Rhizopus species, it's otherwise referred to as the black bread mold. And this is also the most common mucorales member to cause human disease. Next question, which of the following is one of the 4 causative agents of aspergillosis? The correct answer is letter A, diba? So dito po, napakita na natin to kanina, it's Aspergillus niger. Next question, the most common mucorales member to cause human disease is Rhizomucor variabilis, Mucor irregularis, Rhizopus stolonifer, or Leucemia corymbifera. The correct answer is Rhizopus stolonifer. So, ayan po, nabanggit na rin natin ito sa previous slide. The most common mucorales member to cause human disease belongs to the Rhizopus species. So, among these choices, ang pipiliin natin dapat ay yung Rhizopus stolonifer. Keep in mind though na yung nandito sa listahan, ito rin naman po yung 4 na fungal genera na under ng Mucorales, no, ng taxonomic classification ng Mucorales, that's commonly observed to cause human diseases. No? If you would review your Mayhon 7th edition, mahaba, di ba? Mahaba yung listahan ng iba't ibang mga fungal genera that's capable of causing opportunistic fungal infections that is classified under Mucorales. Pero may 4 po na madalas na ma-associate with human infections. And yun nga po yung Rhizopus, Mucor, Rhizomucor, and Leucemia, which is formerly known as Absidia. But again, out of those 4, yung Rhizopus po, yung most common talaga to cause human infections. Next question, which of the following shows the smooth, ovoid sporangium of Leucemia species? The correct answer is letter B. Alright, so as previously mentioned, Leucemia is formerly known as Absidia. Leucemia is also a Mucorales member, although it's quite less common kung ikukumpara natin sa Rhizopus as an isolate or as a causative agent of human infections. So we recognize the presence of Leucemia in samples by observing their very smooth and ovoid sporangium, as we can see right here in this example. Next question, based on colony morphology, identify the fungal organism shown here. Our choices are Aspergillus flavus, Cunninghamella, Geotrichum candidum, or Candida cruzae. The correct answer is Cunninghamella species. So we recognize the presence of Cunninghamella species if we observe their colony morphology, because their colony morphology has been described to be white and cottony. Okay, mukha daw pong bulak yung colonies ng Cunninghamella species. But keep in mind though that the colonies of Cunninghamella eventually begin to turn gray as the colony ages. Okay? So habang bago pa po yung isolate ninyo, habang bago pa yung culture ninyo, they appear to be white and cottony. Pero habang tumatagal po yung incubation, nagmumukha daw po silang gray in appearance. Alright? But again, no, it's quite notable that Cunninghamella species is capable of producing white, cottony colonies. Next question. Identify the fungal isolate shown here. Our choices are Aspergillus fumigatus, Terriens, Niger, Flavus, or Glaucus. The correct answer is Aspergillus terreus. So we've already seen this slide in the previous question, diba? So Aspergillus terreus, it's capable of producing that kind of like dusty pink or old pink colonies with kind of like a tan or beige outer edge. Tignan po natin tong picture na to, no? So parang may dull pink appearance dito sa gitna, but on the outside, no, they're capable of producing, ayun nga, this tan, light tan or beige color sa paligid. So again, pag nakita tayo ng ganito, we're looking at Aspergillus terreus. Next question, which of these photos is showing us the colony of Aspergillus flavus? The correct answer is letter C. Diba? So as previously discussed, if the organism produces Pink colonies with a light beige outer edge, that's Aspergillus terreus. Dito naman sa B, dark green colonies with a white outer edge. This is Aspergillus fumigatus. And as you can see right here sa option C, bright yellow or bright yellow-green colonies would indicate the presence of Aspergillus flavus. Next question. Identify the organism shown here. Our options are Aspergillus fumigatus, Syncephalastrum racemosum, Penicillium citrinum, or Purpureacillium lilacinum. The correct answer is Syncephalastrum. So Syncephalastrum is also a mucorales member but is only associated with opportunistic infections. All right. Now the problem with Syncephalastrum species, it's sometimes they may be confused with Aspergillus species. All right. But again, on the previous question, it's showing us a photo of Syncephalastrum. Next question. Which of the following is the causative agent of mycotic keratitis? The correct answer is letter A, Fusarium species. So Fusarium species is also listed as one of the causative agents of opportunistic fungal infections. But ayun nga po, Fusarium has been known to cause mycotic keratitis among soft contact lens users or if they're using contaminated lens solutions. This may also happen if a person wears their contact lenses for days without removal, which is not recommended. Diba? Dapat less than 8 hours a day lang yata dapat yung pagsuot ng contact lenses. So, of course, consult with your doctor before you actually proceed to wear contact lenses para po mabigyan kayo ng tamang instructions on how to properly wear your lenses and kung gaano lang dapat katagal nasuot yung contact lens. Because, again, they could lead to eye infections such as mycotic keratitis. So how do we recognize the presence of Fusarium species in our sample? Upon microscopic morphology, we should be able to observe canoe-shaped or banana-shaped macroconidia. When we say canoe, hugis, bangka, canoe are types of boats. Okay, it's a type of boat. When we say banana-shaped, ayun nga po, as we can see right here in these examples, they are quite long and curved. Diba? So, ganun po yung itsura ng Fusarium species. Alright, so again, if we're able to observe this appearance of macroconidia in our sample, that may indicate that we are looking at Fusarium species. Next question. Aside from infections caused by Malassezia for fur, infections by which other organism may we be able to observe using the wood lamp? Our choices are Fusarium falciforme, Exophiala ginsulme, Alternaria citri, Candida albicans, or Microsporum oduinii. Alin daw po sa mga ito yung pwede rin nating ma-observe gamit ang wood lamp or woods lamp? The correct answer is Microsporum oduinii. So as you can see right here, aside from Tinea versicolor, aside from superficial fungal infections, pwede rin po natin gamitin yung wood lamp or woods lamp to observe cutaneous fungal infections. And kasama na nga po doon yung infections with Microsporum oduinii. Now, if you would remember from the previous question, Microsporum oduinii is a causative agent of what disease? Gray patch ringworm, diba? Gray patch ringworm, which is a form of Tinea capitis, diba? So, pwede rin po nating gamitan ng wood lamp or woods lamp yung pasyente para hanapin yung areas kung nasaan may Tinea capitis yung pasyente. And again, one of the causative agents of Tinea capitis, specifically the gray patch ringworm, is Microsporum oduinii. Next question. The best site to collect skin scrapings is at the center of the lesion because this is where the infection started and has the most amount of fungal cells. Yes or no? The correct answer is no. So for the collection of skin samples, they are usually scraped from the outer edge of the lesion. Now, the rationale behind this is dun po kasi sa center ng lesion kung saan nagsimula yung infection, usually po no, that's already heavily clustered with excessive fungal elements and the dead skin cells of the patient, which is why hindi ito okay na source of sample. We prefer the outer edge of the lesion dahil sa outer edge of the lesion po, dito pa lang nagsisimula na mag-spread yung infection. So, hindi pa ganun karami yung dead epithelial cells and there's just the right amount of fungal cells on that area. Which is why, again, the answer to the previous question is no, because the correct area to collect your sample would be from the outer edge of the lesion. Next question, which of these statements is correct? Statement 1. The skin must first be cleaned before collecting skin scrapings to minimize the presence of bacterial contaminants. Statement 2. Use 40% isopropyl alcohol to kill bacteria without the destruction of fungal cells. Our choices are, both are correct, both are wrong, only one is correct, or only two is correct. The answer is, only statement 1 is correct. Let's discuss. So this is also the previous slide that I've shown in the question before this. So aside from collecting samples from the outer edge of the lesion, it is also recommended, or it's part of the protocol, to first clean the skin with 70% isopropyl alcohol before sampling. Okay? So dapat po 70% alcohol solution ang gamitin natin. Mali po yung 40, okay? Hindi 40 lang. Walang gagawin na kahit anong disinfection or decontamination ang 40% lang na isopropyl alcohol. So again, the recommended percentage is 70. Next question. Which of these is the septic check system? The correct answer is letter C. Yung option A po, that is the API for carbohydrate assimilation. Yung letter B, that's the chromogenic candida agar, and the correct answer nga po is letter C. So the septic check system formulated by Becton Dickinson Diagnostic Systems, that is a biphasic, system that makes use of both broth and agar. Okay? So when we say biphasic system, as we can see right here in the sample, yung agar, may agar strips sila dito sa taas, tapos yung broth nandito po sa ibaba. Alright? So what would happen here is you would take your sample, no? Let's say, ang kinolekta nyo po ay blood sample, no? You would take your blood sample, i-inoculate mo siya mula dun sa taas. Now, from the top, the sample would flow towards the bottom. So, yung sample po, dadaan muna sila dito sa agar strip na nasa taas, diba? So, dadaan muna sila dito and the agar would be inoculated by the sample. And then eventually, the sample would flow downwards and mix together with the broth at the bottom. Okay, so ulitin po natin, the septic check system is a biphasic setup wherein there is an agar at the top and there is a broth at the bottom. So when you inoculate your sample, it flows through the agar at the top and then mixes with the broth at the bottom. So again, that's the septic check system. All right. Next question. Based on the Chrome-agar Candida sample shown here, identify which would be the growth of Candida tropicalis. A. Purple-colored colonies. B. The white-colored colonies. D. The light blue-colored colonies. And E. The dark blue-colored colonies. The correct answer is letter E. So as you can see right here, ito po yung iba't ibang mga Candida species and their appearance on the chromogenic agar Candida. So the light blue colonies are produced by Candida albicans. The dark blue colonies are produced by Candida tropicalis. The white colonies is produced by Candida glabrata. The purple-colored colonies is produced by Candida crocei. And if you're seeing a metallic blue colony on your plate, then that is Candida dubliniensis. Now, on the previous photo, ang pinakita po natin ay Candida tropicalis. Here are all the references that were used in the making of this discussion. And that ends our summarized review discussion in mycology with practice questions. This has been Aurel Enriquez. Thank you so much for watching.